Good afternoon, students. Welcome to class 10. Okay. The last video I have just started the heredity and genetics. Today I am going to start the Mendel contribution. Very important, very much important. Mendel contribution and Mendel law. Actually, Mendel is known as a father of genetics. He had a great interest in the plant breeding, hybridization. He had great interest, and he conducted many of experiment. After that, over 8 years, okay, and he selected pea plant. Actually, he selected pea plant, and after experiment, he published. Actually, he published the experiment, the common. The published after experiment, he published the conclusion. Okay, and conclusion of his famous experiment on the common pea plant. And before the field of science, the genetics did not begin until 1866. After that, when Mendel concluded his experiment, when he conducted experiment, the genetics started to study all the human being know about the genetics. That's why the Mendel is known as a father of genetics. Okay, the first. Actually, he selected the pea plant. Why he selected pea plant? Why? Others plants, why not? Okay. He selected the pea plants. The pea plants have the several varieties, several distinct characteristics having in the both pea plants. And pea plants, another point, the pea plant have the bisexual, having male and female. Okay, all are the experiment for this, and also in a pea plants, due to self-fertilization, it is easy to get pure lines of several generations. In the pea plants, when you do the self-fertilization, the self-pollination, easily he got a pure character, and the pea plant also a short lifespan, and he possible to study the several generations within a short span of time. Okay, this is the experiment. Actually, was the experiment on a pea plant. Actually, the main experiment, and this is experiment including the 3 stages. The first, but he selected. Actually, this is the first stage. The first stage is experiment. He conducted experiment in the 3 stages. And in the first, the Mendel selected the 7 pair of contrasting characters. 7 pairs of different characters. Okay, 7 pairs characters given in your book. Just read the character, the plant side tall and short, green, yellow. Just read this. What is the dominant character and what is the recessive character? I am not going this type of characters. Only I am discussing. This is the experiment, Mendel experiment. This experiment, actually the 3 stages. In this conducted his experiment, there are 3 stages. First, the 7 pair of contrasting characters, and he self-pollinated the pea plant for the several generations. Self-pollinated. Actually, is allowed to self-pollinate the pea plant, and he got he observed that seed from a tall plant or the produce a tall plant. And on the seed from a short plant, this seed produce only short plant. Okay, and seed, suppose this is the purple color flower, the seed from this plant, the seed produce only purple color flower. This is the first experiment or the first stage. Stage 1. Okay, first experiment. He is allowed to do self-pollinate. The stage 2. This experiment is very much important. Stage 2. He crossed 2 plants. And the contrasting characters means different character. But obviously, this is the pure character, not hybrid. Okay, pure character means in the stage 2, he crossed 2 plants. Suppose T and T. The tall and short. T and T. But this is pure. Pure means this is the condition is the homozygous. It's a pure, homozygous, not heterozygous. Okay. They are tall and short. He crossed between two constituent character of the two plants, and in the this is called the parents. And then here the gamete. From this, you know, gamete is produced, T and T, and this is T and T. Okay, this gamete. And he observed that he got the plant from this after cross pollinated. Okay, but this plant with the TT and again T to T, TT, again this T to T, means 4 this type of T and T. But this offspring is phenotypically showing is a tall plant, but genotypically this is the hybrid. Okay, this is the hybrid plant. Genotypically it is the hybrid, but its condition is the heterozygous condition. But this phenotypically showing is a tall plant, and this cross is called monohybrid cross. Monohybrid cross means in the characters the one characters. So it is called the monohybrid cross. When the two characters called a dihybrid cross is the monohybrid. Only one character is there. This is tall and the short, and then we get the offspring. He got offspring. The offspring of the tall, the plant. This plant, the tall, phenotypically showing tall plant. Okay. The stage 2 is experiment. Stage 2. After that, the stage 2 experiment. This generation. This means the this generation is called the F1 generation. F1. Okay, find it. F1 generation or the first generation is called the F1 generation. After that, stage 3, he allowed to do the self-pollinated among this plant. Okay, this TTT we got, whatever you got, is allowed to do self-pollinated. Self-pollinated means obviously the T, the T. This is called the self-pollination. But this is the F1 progeny, F1 generation. Gamete will be the T and T. This is the gamete, T and T. This is gametes. And he observed the plant, produced the plant, because if you cross this from this to this, T, after this cross T, this, T, and T, and T. See, whatever offspring he got, T, and the last one you can see this, the short plant. In this generation, or this is the F2 generation, he got short plant. Okay, and also the you see all the tall, but phenotypically it is showing tall, but genotypically it is the different. Okay, T, and what is the ratio of this? Ratio is the genotypically the offspring TT1, T, capital T, small t, T, and TT. The what is the phenotypic ratio? Phenotypic ratio, phenotype means the visible characters. Visible characters, see, all are visible characters, all are tall. Okay, means 3. But here only one plant. This is short plant. This character, the phenotype, and the genotype. Genotype, see, genotype means it's a homozygous or heterozygous. See here, the one, but this is the tall. Okay, one. And this is genotype, the same genotype. Two, the genotype is different. This is the ratio. Very much important. This is the dihybrid cause. This Mendel, the first experiment is the called dihybrid cause, and the phenotypic ratio is the dihybrid cause. 3:1. Genotypic ratio, this is the first cause is monohybrid cause. Okay, the phenotypic ratio of the monohybrid cause 3:1, and genotypic ratio of monohybrid cause 1:2:1. Okay, 1:2:1. This is the monohybrid cause. Monohybrid cause means the one characters is there. The different characters, one character, not two characters. This is tall and short. That's why it is called the monohybrid cause. The monohybrid cause, the phenotypic ratio 3:1, genotypic ratio 1:2:1. This is the first experiment, the Mendel's experiment. Very much important. After one experiment is the dihybrid cause. Next video I will discuss. After that he conducted law, the Mendel's law of inheritance. Okay, this is the monohybrid cause, and this is the this is the F2 generation. F2 generation offspring, and he got this one. Only one he got short plant, but he got short plant. Okay, but first generation you did not get short plant. Okay, only tall plant, but this is the hybrid. Okay, and phenotype is 3:1, genotype 1:2:1. Genotype means this is the homozygous condition or the heterozygous, but same, the tall. And this is the homozygous, but this is the recessive character. Here is a dominant, this is a recessive characters. But phenotypically visible characters all are tall. That's why here 3, there only 1, 3:1. This is the monohybrid cause. I hope clear. Next video I will discuss dihybrid cause. Okay. After that, Mendel's law. Prepare at home. This chapter is very much important. Okay, and the little interesting chapters. Okay. Thank you.